Okay, so for the benefit of the journalists who are new to the focused ultrasound world, I'll give a brief précis on the technology and on the foundation. So, focused ultrasound is the most powerful sound you will never hear, but it's a sound that could someday save your life. Focused ultrasound is an early stage, totally non-invasive therapeutic technology that could be a highly disruptive, game-changing alternative or supplement to traditional surgery, radiation therapy, drug delivery, and so on, and thereby provide the potential to transform the treatment of a whole variety of serious medical disorders, including Alzheimer's and Parkinson's disease, and OCD and depression, and epilepsy, and benign and malignant tumors of the brain and breast and prostate, and so on, and thereby improve the lives of millions of people around the world. Focused ultrasound has the potential, and I emphasize potential because it's still early stage, it has the potential to revolutionize therapy to the same degree that MR revolutionized diagnosis. But in its evolution, it's where MR was 30 or 35 years ago. Nobody had ever heard of it, and focused ultrasound has been called medicine's best kept secret. So the way it works is analogous to using a magnifying glass to focus beams of light on a point and burn a hole in a leaf. And to simplify, with focused ultrasound, instead of an optical lens focusing beams of light, an acoustic lens can be used to focus multiple beams of ultrasound energy on a target deep in the body with a high degree of precision and accuracy, sparing the adjacent structures or normal tissue. And at that focal point where the beams converge, just like where the beams of light converge and burn the hole in the leaf, at the focal point where the beams of ultrasound energy converge, there are 18 ways in which the ultrasound interacts with tissue, 18 biological mechanisms, in contrast to radiation, which only has one mechanism of action. The fact that there are so many different mechanisms of action creates the opportunity to treat an enormous variety of serious medical conditions. And today there are more than 100 medical disorders in various stages of development, mostly early. For reference, 10 years ago, there were only 3. So it's early stage. There's an enormous amount of work that remains to be done before focused ultrasound can achieve its vision or potential of improving the lives of millions of people. And the problem is that the evolution of any new therapeutic technology, like focused ultrasound, from idea to widespread adoption or diffusion as a standard of care, is a glacial process that often takes decades. So, saving time, saving lives. Every day that there's a delay in the availability of this technology translates into unnecessary death and disability and suffering for countless people. So 12 years ago this month, we created the Focused Ultrasound Foundation, which is a unique medical research, education, and advocacy organization with a simple mission of accelerating the development and adoption of focused ultrasound, saving time, saving lives. And today, as Jessica said, we're going to highlight 2 of the most exciting areas: blood-brain barrier opening and cancer immunotherapy. And we'll start with Nathan.
Thank you, Dr. Meng. Good afternoon, everyone. I'm going to Thank you, Neil. My name is Nathan McDonald. I'm a physicist. I work at Brigham and Women's Hospital on developing new uses of ultrasound from preclinical studies to patient studies. I'm going to talk to you today about one application that we've been looking at for over 15 years now, which is to use ultrasound to enhance the delivery of drugs to the brain. I think there should be some. No. Okay, so if you're not familiar with the blood-brain barrier, in most of the vessels in your body, if you inject a drug or if you take a drug, it will get out of the blood vessels into the tissue. But the brain is different. It's protected. And this is performed by the so-called blood-brain barrier. And this is both a physical barrier that prevents drugs from going through gaps between the cells. So there are these tight junctions that prevent passive diffusion of drugs into the brain. It's also a functional barrier. So it has various mechanisms that prevent or regulate how much what goes into the brain and what's pumped back out into the blood vessels. This is a sort of a famous picture of a mouse, and in this mouse was given a tracer. And you can see that everywhere that is dark is where the tracer goes, except for in the brain and the spinal cord. And this is because of the blood-brain barrier, which is great for protecting the brain, which is very important. But it's also a big problem if we want to develop and use drugs in the brain. So more than 98% of drugs, small molecules, and practically all large molecule drugs do not cross the blood-brain barrier and cannot be used effectively in the brain. And for this reason, it's a major limitation for the effective therapies for brain tumors, neurodegenerative diseases, and multiple other disorders of the central nervous system. So what we work with is ultrasound, and it's been known for a long time, for over 50 years, that sometimes you can apply ultrasound and you don't get any damage like we do with ablation, but you simply see an increase in the permeability in the blood vessels in the brain. And what we worked out was that this was due to microbubbles. And if we inject microbubbles before we apply the ultrasound, that we can concentrate the acoustic effects of the ultrasound onto the blood vessels in a very gentle way. And through some mechanism that we don't fully understand, we get an opening of the tight junctions and we get an increase in active transport. So we see a disruption of both the physical blood-brain barrier and the functional blood-brain barrier. And this works through the microbubbles because they respond very strongly to ultrasound. They're actually used, they're FDA-approved agents for ultrasound imaging. And when they react to the microbubble, you get a variety of mechanical stresses and forces on the blood vessels that somehow result in this opening. And this small movie on the right is from a sort of another famous movie from a group in Seattle. And this is an in vivo image of a microbubble in an ultrasound field. And you can see that it responds very strongly, and you can see that the vessel is enlarged and contracts. And it's through these stresses that we get the blood-brain barrier opening. So these are some examples from the preclinical work that we've done in small animals. So we've been looking at this primarily the first application for brain tumors. So this is a rat, and you can see that we disrupted the blood-brain barrier in the tumor and in the surrounding area. The idea here is to enhance the amount of drug that gets into the brain tumor and to go after these infiltrating cells that really cause the recurrence and the problem in the periphery. So they're impacted by the fully intact blood-brain barrier. The center image is from a mouse that is a model for Alzheimer's disease. This was one of the early studies that was done. And you can see the green area is where we disrupted the blood-brain barrier, and that is where it's sort of diffusely open. And then we injected an agent that it goes after that targets plaques. So you can see these red spots. So these are the amyloid plaques in Alzheimer's disease, and they're lighting up. So this just is a demonstration that we can administer a drug and get them to the plaques. And this is really a drug-neutral approach. So even large things like stem cells, so the right image, we gave fluorescent stem cells to a rat, disrupted the blood-brain barrier, and you could see that we can deliver them. And this may be very important if we want to use stem cell therapies in the brain by avoiding having to directly inject cells. So where are we now? What is the state of the field? This is a reversible effect. So we open the blood-brain barrier for a few hours. It closes exponentially with a half-life of 1 to 6 hours. We've shown that it could be safe, so we can do it repeatedly without significant pathology or any functional deficits. It's controlled, so we can control not only where the barrier is open, so we can direct the delivery of drugs to only the areas that we want, but we also can use feedback control to make sure that it's safe and potentially even control the level of the opening, so we can maybe have an idea of how much drug we want to get to the area of the brain and control that. And as I mentioned, this is drug neutral, so it ranges from small molecules to nanoparticles, liposomes, even cells that we can deliver across. And then finally, it's been shown now that it's been clinically translated. There are human trials now with two devices from 2 companies, and that's what we're going to hear about next from Dr. Lippin.
Have to go forward? Okay, it's counterintuitive. Okay. Great. Okay, thanks very much, Nathan. My name is Neil Lipsman. I'm a neurosurgeon at Sunnybrook Health Sciences Center at the University of Toronto. So what we're working on, as well as translating a lot of the work that Nathan and our group is doing in Toronto to try to open the blood-brain barrier in various clinical trials, and you'll hear some of those now. But what I will provide is sort of a greatest hits of blood-brain barrier work that's happening around the world. So there's a concerted effort, several labs, several research institutes around the world using this technology and other kinds of technology to open the blood-brain barrier. So I don't have a lot of time, but I did just want to highlight the key work that's being done around the world in this exciting area. So as Nathan said, and just to reiterate, the BBB is a major obstacle. It's a huge obstacle for the delivery of brain therapeutics. So there may be effective drugs, there may be effective agents for many neurodegenerative and other disorders, but we just can't get them into the brain in sufficient concentrations to have a meaningful impact. So overcoming the BBB has been a major goal for several decades. It's been a holy grail of sorts. How do you safely and reversibly open the BBB in a way that can close afterwards but can also deliver something in an image-guided, ideally fashion? So approaches to date, so people have tried other kinds of things. Approaches to date have been limited, though, by safety concerns. So the techniques that have been tried maybe aren't so safe, relatively speaking. Inefficient or insufficient delivery, there may be ways to do it, but just you can't get enough of the compound to the brain. And expense, there may be expensive or other logistical challenges. So as mentioned, a safe, reversible means of opening the BBB directly at the site of pathology, so directly where you want to deliver something, has been an important goal for the field. What are some of the priorities? Neurodegenerative diseases, cancer, stroke, the conditions that impact humans around the world in large proportions. So if you look at the World Health Organization list of the top 10 most common diseases to affect human beings, these are going to be at the top. And so these are the priorities for the field. So they're common, they're deadly, and there have been little clinical advances in the last several decades. And there is a therapeutic gap in the field. So what that means is that we have advanced our ability to diagnose a lot of illnesses, in the last 20 or 30 years, but we have not been able to advance our care of these illnesses. Alzheimer's, glioblastoma are good examples of that. We can recognize what they look like under a microscope, genetically, et cetera, but we just have not been able to make that leap to treat them and to prolong life. Okay, what did I do here? Okay, it's upside down. Good, okay. So the symposium that we're at right now gathers work from institutions across the world. It's a unique opportunity to really have everybody in the same room under one roof to really discuss different techniques, different populations, but all guided by the same question. Can opening the blood-brain barrier in human patients be done? Is it possible to do it? Is it safe to do it? And is it effective? Okay, this is a graded sort of process where you have to answer one at a time in a rigorous, scientifically sound way. And it is not a trivial question. So the blood vessels that we're talking about are very small, and in some of these populations, patients are sick. So it's not a trivial question to establish that, in fact, it is possible to do it transcranially without opening the head and to do that safely. So again, go through some of the highlights of the work. So this is work that we did in Toronto in the context of Alzheimer's disease. So Alzheimer's disease, obviously, most common neurodegenerative illness, one where many billions of dollars have been spent on trying to develop antibodies to get into the brain, and so far the blood-brain barrier has been a major obstacle. So what we did is we opened up the blood-brain barrier in Is there a laser here? Or if you look at the, sorry, it's not pointing at the thing. Okay. In any case, we managed to safely open the blood-brain barrier in 6 patients with early to mid or mild to moderate Alzheimer's disease. These were patients that had amyloid positivity on preoperative amyloid scans, which means that the areas that we were opening were positive for amyloid. And this was buoyed a lot by preclinical data suggesting that opening up the blood-brain barrier may potentially be useful in clearing amyloid. But what we found was that you can open the blood-brain barrier repeatedly. You can do it reversibly, so it was closed the following morning when we tested, and you can do it again a month later in a larger volume. So it's repeatable and reversible, and it looked to be safe. So this work is now continuing to larger trials. Work done by Dr. Price in Parkinson's disease. Here, I'd highlight this, several unique aspects of this work. So this is in the context of Parkinson's disease in a preclinical trial, so an animal model trial of focused ultrasound-mediated blood-brain barrier opening for the delivery of GDNF or gene for GDNF in Parkinson's disease. So there has been a tremendous interest in trying to deliver growth factors to the brain in patients with Parkinson's disease. So far, human trials of inserting cannulas into the brain and directly delivering this have failed. A large consensus is emerging that they may have failed because just not enough of that is getting into the brain. So what they were able to demonstrate is a successful and significant delivery of GDNF, of this growth factor gene, into the brain, and very importantly, an actual change in behavior. So when you look at these animal models, these Parkinsonian phenotypes, Parkinsonian behavior, they're actually able to be reversed with this. So not only can you deliver it in sufficient concentrations, you can actually get reversal of some of the effects. Moving on to brain tumors in glioblastoma, this is work again done at Sunnybrook, and for some reason the images do not show up there. Essentially, what we've done is we've started and launched a phase 1 trial, a proof of concept trial that you can open the blood-brain barrier in patients with glioblastoma, so the most malignant, most common, and aggressive brain tumor. The goal there is to enhance the delivery of chemotherapy to the rim of the previously resected tumor, because that's where 98% of recurrences occur. You may be able to take the tumor out, it recurs invariably. And when it does recur, it recurs in the 1 centimeter margin of the place that you resected, because it's impossible to resect every last cell of this tumor. So if you can enhance the concentration of chemotherapy delivered to that part, you may have a chance of altering the natural history of that tumor. So what we did is a small trial in 5 patients to demonstrate that it is in fact safe to open the blood-brain barrier in this population. And in fact, we managed to do that with no adverse effects. So we can actually open it, it's reversible, and we can do that in a tolerated fashion. Oh, there it is there. Okay, good. So so I'll just draw your attention to the MRI scan on the left there. The patient has had a tumor resected from the right frontal lobe. That's the sort of the empty spot there. And on the right, you can see immediately behind that a fluffy white contrast. And that's the area that we are able to open the blood-brain barrier in. Okay, so again, that area is exposed to a significant increase in concentration of whatever the patient is taking, whether it's temozolomide or another chemotherapy agent. So we're able to do that reversibly. So moving to recurrent glioblastoma, this is a group led by Dr. Lung. It's a different kind of technology. So we use in Toronto Insight Exablate machine. This is another kind of technology that utilizes intraoperative neuronavigation guidance, CT-based treatment planning, and a technique that is streamlined for multi-course administration. So here what they were able to show, and they treated their first 2 patients in August of this year, successfully, and they're able to show that they were also able to get positive contrast enhancement indicating BBB opening with a different kind of device. So again, underscoring the fact multiple centers around the world utilizing different technologies to achieve the same kind of objective. This is work done in Paris by Dr. Carpentier, who's here. It's a very large experience by Carthera using low-intensity ultrasound again for transient BBB opening in patients with recurrent GBM. So this involves a surgically implanted transducer. So these are patients that have a recurrent glioblastoma, and at surgery they have implanted a transducer. And that is in anticipation of a 10-minute procedure, and they've treated several patients to date. to open up the blood-brain barrier in these patients. And they're providing very compelling data indicating that it's safe and may, in fact, influence overall survival in these patients. So again, they get ultrasound in combination with chemotherapy. This is work done by Dr. Woodworth, who's here. So the first patient in the United States to have a blood-brain barrier opening for tumor was done just a few days ago, actually, successfully. And again, drawing your attention to the circle indicating an area of contrast enhancement. So again, you can't really underscore or underemphasize the importance of seeing that contrast enhancement, which indicates that that part of the brain is being exposed to significantly increased concentrations of whatever therapeutic compound it is that you want to deliver. Okay, the fact that that is reversible, the fact that that is safe and temporary is a key step forward. And that trial is currently ongoing. Moving to Parkinson's disease now, again, this is work by Dr. Zhang, again, a preclinical trial of ultrasound-mediated BBB opening for delivery of anti-alpha-synuclein antibodies. So we know that alpha-synuclein is a pathologic protein that occurs in patients with Parkinson's disease. It's a hallmark, a pathologic hallmark of PD. If you're able to deliver an antibody to that, similar to anti-amyloid antibodies in Alzheimer's, then you may be able to have an impact on Parkinson's disease. One of the challenges with Parkinson's disease is that all of the treatments that we have to date are symptomatic, which means they treat the symptoms, but they don't alter the natural history of Parkinson's, which has been a major goal for the field, to try to influence that invariable decline. So what they were able to show in 10 transgenic Parkinson's mice, they're able to open the blood-brain barrier effectively. They were able to deliver significant concentrations of antibody, and they were able to have a significant influence on alpha-synuclein. So they can deliver it safely in these animal models, and they can actually influence alpha-synuclein concentrations. So this is really important work that's ongoing. So the hope is obviously able to translate this to humans. Now, if we look at the animation that Nathan showed, an antibody is several thousand times larger than the typical compound that can cross the blood-brain barrier. So if you have something that you can cross, for example, glucose or water, et cetera, an antibody is going to be thousands of times larger. So if you can show that you can get that across, then really, all bets are off. You can really cross virtually any compound that you want to deliver. Stroke as well, I mentioned this is work being done by Dr. Kalani and demonstrated that you can open the BBB with focused ultrasound after an ischemic stroke and deliver mitochondria. So mitochondria are the factories of the cells. They help derive energy for the cells. So you can actually, and they were able to show that in a middle cerebral artery occlusion model, they were able to tag mitochondria, and you can see all those red spots there in the bottom indicating that mitochondria was able to deliver to the area of a previous stroke. So even strokes, even conditions where previously thought we cannot really touch, that as soon as somebody has a stroke, they're sort of done. Focused ultrasound may play a profound role in delivering healthy things to the brain. So really, that's just really a greatest hits collection. Sorry for the speed, but really just wanted to give a sense of what kind of work is being done around the world, different centers, different technologies. Where are we going now? Certainly, this is our early days, as Dr. Cassell mentioned, but there is intense interest in improving every aspect of the process. So improving the technology, both the hardware and the software. Currently, we use stereotactic frames that are attached to the patient's head to keep the head perfectly still during the procedure. We are headed towards a frameless era. Image guidance is improving as we get much better resolution imaging. Both structural and functional imaging will enhance our ability to target the brain. Delivering effective therapeutics. So as we work on focused ultrasound, there's a whole other groups of scientists around the world working on developing what it is that we want to deliver into the brain. So novel biologics, novel treatments, being able to radio label potentially compounds so that we know that we're delivering what it is that we want to deliver. That's a key aspect of this technology. And finally, really optimizing patient selection. So we know that, patients succeeding and patients doing well, a lot of that has to do with selecting the right patients for these procedures. And for that, all of this work is incredibly multidisciplinary. We work very closely with our colleagues in other disciplines to move this forward. It's one of the beauties of working in this area of clinical trials, that you work closely with your colleagues in this area. and really, in general, the movement towards intervening earlier in these illnesses. So whether it's Alzheimer's, whether it's brain tumors or other indications, the earlier intervene, we believe, the better chance we have at making a difference. Okay.
So thank you, Nathan. Thank you, Nir. And now I'd like to also welcome to our panel Q&A discussion Dr. Alexander Carpentier, Dr. Jürgen Getz, and Dr. Richard Price. And so we now have time for Q&A. First, if there's anyone in the audience who has a question, please put your hand up. Okay. Do we have any questions from online?
I have one question that's come in.
Okay.
We saw media coverage last week that the first U.S. patient with early Alzheimer's was treated at West Virginia University using focused ultrasound to open the BBB. What can you tell us about that treatment and that patient and that type of?
Well, we actually are fortunate to have Dr. Ali Rezai from West Virginia who treated that patient. Perhaps he'd like to share.
Yes, so this is in collaboration with other centers. This is a Incytec sponsored FDA phase 2 trial looking at safety and clinical benefits and also reduction in plaques. So last week we performed the first procedure in a patient last Tuesday at the Rockefeller Institute who was a 61-year-old female named Judy, who is a PhD in nursing as an instructor for the neonatal ICU and practitioner who had to stop working because of her early Alzheimer's. And she underwent a procedure last Tuesday. We targeted the hippocampus and the entorhinal cortex, two targets that were rich in plaques using PET scanning. And Judy tolerated the procedure well. The BBB was open for 48 hours. And she went home the day after the procedure, and she's so far stable neurologically with the first treatment.
Thank you.
We've got 2 more to go. Every 2 weeks we do the procedure. Next one will be next Tuesday, and then following 2 weeks after that.
Great. So this is really exciting to hear about these different clinical trials ongoing for Alzheimer's. So I know that we have Jürgen Getz on the panel, and he completed some of the seminal preclinical work that really helped pave the way for these clinical trials. So Dr. Getz, would you comment on that work and then what you're doing now in the preclinical realm to really try to understand what capabilities focused ultrasound has in treating Alzheimer's?
Mm-hmm. Absolutely. So we have started initially treating an Alzheimer's mouse model with an amyloid pathology. And maybe it's worth comparing the A-beta and the tau pathology. As you know, A-beta is deposited extracellularly, whereas tau, similar to alpha-synuclein, forms lesions intraneuronally. So obviously, A-beta and tau present different targets. And for A-beta to be cleared, any therapeutic agent needs to reach the interstitial space of the brain. But for tau or alpha-synuclein to be cleared, obviously, the therapeutic agent or whatever is entering from the blood into the brain needs not only to enter the interstitial space, but also needs to enter the neurons where all the damage occurs. So in the case of the A-beta depositing mice, the mice with the amyloid pathology, we were sonicating the mice using a scanning ultrasound procedure where we targeted the entire brain. And we did this for either we had between 4 and 7 weekly sonications. And even in aged APP mutant mice with an amyloid pathology, this was sufficient to reduce the amyloid plaque load by approximately 2-fold. And in the case of a younger cohort of mice where we treated between 5 and 7 times, we were able to reduce, again, the amyloid plaque load by approximately 2-fold, and at the same time, we were able to restore memory functions fully. And then we moved on to the tau pathology, using a tau transgenic mouse model, where we treated the mice 4 times, and we had 4 treatment arms. So we had a single-chain antibody variant against a specific form of tau. We used ultrasound on its own, the 2 in combination, and we had a no treatment arm and where we found that 4 treatments were sufficient to reduce tau pathology to some degree, but also, as far as memory functions were concerned, it needed a combination of a single-chain antibody variant and the ultrasound treatment. And we have since moved on to a tau transgenic mouse model with a motor impairment, doing 14 consecutive treatments, and where we found that ultrasound treatment in itself was sufficient to ameliorate the motor phenotype. So to me, it seems that when you want to target A beta, ultrasound on its own will do it. Possibly when you want to target tau, either you have to do more treatments or you have to use, in addition, a therapeutic agent.
I'll just add, just as a comment, I think that underscores, in the context of BBB, the power of ultrasound, because it is agnostic to what it is that you want to deliver. So I think that there's not really, as a neurosurgeon, an analog for that. So that's why I think, Jürgen's work is so powerful, because it suggests that, for amyloid, you may be able to use ultrasound on itself, by itself, to help clear some of the amyloid plaques, but for tau, you may need to deliver an agent. So it's not sort of a one-trick pony kind of thing. It's there's multiple kind of mechanisms at play. So I think that's what really underscores the universality of it.
So in thinking about delivering the agent for Alzheimer's, I know specifically there have been many clinical trials recently that have failed for different drugs. Have those primarily been, or what are the reasons? Is it because of not getting through the BBB? Is it not reaching the right target? Is there anything there that suggests that the capability of focused ultrasound, in addition to some of those drugs that maybe otherwise would be abandoned, would be a good combination?
Yeah, sure.
I think what people say is most likely there has been too little too late. And I think we know that there are now antibodies in phase 3 clinical trials which look promising. For example, aducanumab, which is developed by Biogen. And the phase 3 data will be revealed in, I think, early 2020. But I think one issue is that if an antibody, let's assume an antibody becomes approved, it will be quite costly. So Todd Goldie from Florida provided an estimate that an antibody therapy would cost between $25,000 and $50,000 per patient per year, and that is obviously not sustainable for most societies. But with ultrasound, obviously it's possible to increase the brain uptake, so potentially one can reduce the cost if one were to use an approved therapeutic antibody in combination with ultrasound. And I think this morning the question was raised in the context, I think Dr. Fishman asked this question about alternative approaches, for example, using bispecific antibodies, using the transferrin receptor as a target for brain uptake. And I think there one also gets an increased uptake. But I think ultrasound has much more potential.
Yeah, I would echo everything Jürgen said, and again, highlight the cost aspect of things. So when you look at the paper that was published in Nature by Biogen last year, they were probably able to get, and that was sort of a wild success in the anti-amyloid world, able to reduce amyloid to baseline and also see an improvement in a cognitive measure. And it was published subsequently in Nature, but they were able to get about 1% of antibody, and if you look at the costs of that drug, we're talking about $7,000 or $8,000 a month for those patients. So it is not sustainable for a problem like Alzheimer's disease to have even if it works. We can't use it in large quantities. So as a result, there needs to be, and by the way, that will affect immunotherapy as well. So immunotherapy compounds are also incredibly expensive. So I think any approach that you can maybe reduce costs, I think, is imperative. Certainly, I think Canada is no different from that from anywhere else.
So just before moving from Alzheimer's, what are the next steps clinically that you're planning?
Yeah, I think we, having established or at least collected some compelling, what we believe is compelling safety data that you can do, you can open the blood-brain barrier safely and reversibly in these patients is to expand that and test the hypothesis that that opening the blood-brain barrier alone may be effective for amyloid clearance. But ultimately, I think we are headed towards pairing FUS-BBB opening with a therapeutic. I think it's incredibly important also to study as much as we can these patients, and again, as rigorous a way as possible, and that means collecting biomarker data, spinal fluid, serum, et cetera, to make sure that we're measuring and not missing anything in these patients. And I agree completely about intervening early is important.
Great. So shifting to talk a little bit about glioblastoma, which of course is a huge unmet clinical need, I found what was really exciting is that we heard about 3 different trials with 3 different focused ultrasound devices by 3 different companies in this space. And that's, you know, obviously speaking to the power of the technology and the growth of the technology. So I'd like to hear from Dr. Carpentier, because he has a very specific system that he's using, which I'd like to hear a little bit more about that system and their results so far, and then where they're headed next.
Thank you, Jessica. Yes, as you know, ultrasound has difficulties to cross across the skull. So my neurosurgical vision was to say, well, since for a certain patient we need to operate it on to do surgical removing of the tumor or to do the biopsy, I said to myself, well, why not taking advantage of the disease surgery to implant some ultrasound transducer? And this is how it goes. And we started in 2015 by implanting some little chips in the thickness of the bone, of the skull bone, and we can activate those chips right before the chemotherapy the patient has to receive. And like this, it does increase the drug by a factor of 7 inside the brain and inside the tumor. So by now, we did 21 patients in this trial. This trial is closed. And some patients are still alive 1 year after, although they were supposed to be in recurrence of, second recurrence, because I'm doing this at recurrent, in no more than 2 or 3 months. So we have some good hints, and we have some prolonged progression-free survival, meaning that the tumor is not increasing for a longer time than it should usually do. And the survival seems to be also significantly longer. So what we discovered is the little chips it's pretty small, so it's not addressing all the brain, of course, and not all the tumor region also. What we discovered is that the tumor was not increasing inside the ultrasound field, but when the patient was recurring, the tumor was recurring outside the ultrasound field. So the next step for me is to implement several chips in order to be able to have larger efficacy. Voilà.
Great.
Merci, Jessica.
Great. That's very exciting. And Dr. Price, I know you've also had a lot of experience in preclinical models of glioblastoma and treating in some different ways, both in terms of immune-based treatments, which we'll talk more about immunotherapy coming up, but also in delivery of some sophisticated nanoparticles. So I wonder if you could speak to maybe the future of some other potential things that we could deliver through the BBB.
Right. So certainly, I think this was set up pretty well in a previous discussion that Dr. Lipsman and Nathan were having before, talking about the size of agents that can cross the blood-brain barrier after we open with focused ultrasound. So when we do a treatment, we use a contrast agent, which is relatively small. It's about 1 kilodalton in molecular weight. When we deliver an antibody, that's much, much larger, maybe on the order of about 140 kilodaltons or so, which gives you a molecular, a hydrodynamic diameter of less than a nanometer or so. So we like to do things like really big. So we like to deliver nanoparticles. In our work, we found that we can get nanoparticles on the order of 40, 50, 60, 70 nanometers across the blood-brain barrier in a safe manner. And for us, that opens up a lot of opportunities because we can tune the payload in the particle. We can use controlled release nanoparticles for drugs, or we can do different kinds of gene delivery using non-viral carriers that are essentially in that particular size range. So, broadly speaking, I think that is an opportunity going forward. I think it's extremely exciting right now, the clinical trials using FDA-approved agents. That's exactly the way that this has to be done. This has to be done now. But I think looking 5, 10, 15 years down the road, my hope is that some of these more complex delivery agents can start to be incorporated into these treatments and give us, potentially better opportunities to see efficacy in the future.
Thank you. And, I know we often get the question is, well, why do we want to, why should we open the blood-brain barrier? We've thought that it's there for a reason, and is this really a safe thing? And I know that, Nathan, you've done a lot of work in kind of assessing long-term toxicity of opening the BBB and just other modes of looking at the safety. So I wonder if you could speak to that.
Yes, so we've been looking since we first started at how safe this is that we can do this. And at first we were, getting, maybe a few blood cells along with the disruption, and we would talk to neurosurgeons and we would say, well, if we wanted to use this for brain tumors, they would say, why are you worrying compared to even putting a needle in the brain? This is nothing. But since then we've learned a lot, and we've learned a lot about how we can monitor what's happening during the ultrasound. So we can listen to the sounds coming back from these bubbles, because you put a sound in at one frequency, and when you have a bubble, you get sound back at multiple frequencies. And you can use that to understand what's happening at the focus. So I think now that we really do have a good understanding of how to do this safely, we've seen a number of studies from our group and from others who have done repeated treatments, and we don't see any evident damage to the brain, neuron loss, functional deficits. Obviously, we are doing something because we are putting things into the brain that aren't normally there, and there is an inflammatory response. But from in our experience, that is a temporary effect that goes away, and that actually may be something that is that we can take advantage of and may be useful, and for things like maybe that's involved with what we've seen with Alzheimer's and maybe with immune therapy. But overall, at least in the preclinical studies, and now we're seeing in more and more patients that this does seem to be something we could do safely. And I think for the applications we're talking about, primary brain tumor, Alzheimer's, these patients really have no hope right now. I mean, I think that we could really make a difference, and that doing these is so important to show that it's safe. And ultimately, then we could consider using this for other things that are where there are no other drugs, so psychiatric disorders and other things where you might want to get drugs across the blood-brain barrier that don't normally reach there, or direct them to only the areas of the brain that we want to avoid side effects.
And Dr. Carpentier, do you have anything to add? Because I know in your trials with several patients, you've treated them multiple times as well. So the safety of that kind of repeated treatment.
Yeah, at the beginning when I designed the study, I planned to treat the patient maximum 6 times. It's once a month, actually, because actually those kind of patients, the tumor is recurring at 2 or 3 months only. And the fact is I had to do some modification in the protocol to ask the authorities, the French authorities, to be able to continue the treatment since the tumor was not recurring. So I went up to 12 sessions in one patient. And if you compare the MRI before the treatment and after the treatment, you see absolutely nothing. To be, of course, it was scary to open the blood-brain barrier and to think that many components of the blood can get into the brain. But actually, since it is temporary opening, it's very well tolerated. It seems to be. And in addition, you have a cascade of different phenomena that occurs when you do the blood-brain barrier opening, meaning that you have some cascade phenomenon for at least 15 days after the sonication, although the blood-brain barrier is closed 8 hours after your sonication. So there is a good hope. It's not only to get drugs in, but it also to stimulate, as Nathan say, also the potential of the natural capacity to fight against the tumor. Just to let you know, the brain, your immunity don't go into the brain due to the blood-brain barrier. So you have no defense. If you have a brain tumor, you have very, very low defense to fight against it. And now we know that immunotherapy and immunomodulation is a very new field for all other type of tumors, and very efficient. You're stimulating our own immunity helps a lot, much more than chemotherapy sometimes, helps a lot to fight against cancers. So by having this immunity getting into the brain is, I think, for the future, very, very good hope.
Great. And finally, I'd just like to ask, since at the Focus Ultrasound Foundation, our mission is really to speed up the new applications of the technology and help break down any barriers that we can. Do any of you want to share what you think are kind of the biggest obstacles yet to overcome in this field to help identify, what we need to focus on to help move this along? Anyone?
Certainly from the clinical side, as always, it's funding, and that's where, the Focus Ultrasound Foundation has helped us tremendously to accelerate a lot of this work. So not only for funding for startup trials, but for larger trials. So that's key. A lot of the challenges for a lot of these trials are regulatory in nature as well, getting approval both locally and federally, or permission to actually do these trials. And for that, you need to have a good relationship with the industry sponsor, which we have, and again, we rely very heavily on them. So I think those are the two key challenges. They're obviously overcomeable and navigatable, but it requires a lot of effort and nurturing those relationships.
Anyone else?
So less a suggestion and more of a comment, a positive comment. I mean, one thing that the foundation has already done, which is extremely useful, is that they've, in the immunotherapy space, maybe this is more of a segue into the next section, but it relates to this section by the GBM work that a lot of people are doing now, is the foundation has come forward and funded a lot of studies looking at different kind of energy deposition regimes, meaning different types of focused ultrasound, even beyond blood-brain barrier opening, on the immunological response of these particular tumors. And this has been extremely valuable because it's the kind of sort of descriptive science that the NIH wouldn't necessarily fund, because it doesn't necessarily have a mechanism up front, but it's generating the data we need now to develop the hypotheses to go after the mechanisms. So this particular program that the foundation has had in place has been extremely beneficial to a number of teams, I think. So keep that up.
Thank you. And that's a perfect segue to our next topic, and we'll get back to probably talking just about that and the different mechanisms of ultrasound. So I'd like to thank this panel to, talking about the power and promise of opening the BBB, and we will move along to the cancer immunotherapy panel, and we'll hear from three speakers, Jill O'Donnell-Tormey, who's from the Cancer Research Institute, Kathy Ferrara from Stanford, and then David Brennan from University of Virginia.
Thank you very much, Jessica. So hopefully my slides come up. Okay. So as Jessica said, I'm the CEO and Director of Scientific Affairs at the Cancer Research Institute, and this audience may not know who the Cancer Research Institute is, but we're an organization that's been around for 65 years with always the singular mission of supporting research on the immune system that would ultimately lead to the development of effective immunotherapies. So we are now in kind of this amazing space for CRI as kind of validation of a very long-held hypothesis that people didn't believe, but I think by taking the very long road and always funding excellent science, we can lay claim to having really supported the foundation that is leading to the successes that is taken today. And I think my role here is really to introduce immunotherapy to this audience, which may not be typical. So why is immunotherapy so attractive? And I've just tried to put down kind of key topics of why. I think it's, it's extremely powerful because we've seen that it can transform diseases, cancer into a manageable disease, and that, your immune system can actually be stimulated in a way to outpace cancer growth. It's obviously adaptable. Your immune system is always adapting to new challenges, and this is an extremely important criteria, I think, for developing therapies that can actually deliver durable therapies. We know the immune system has memory, and I think being able to generate T-cell memory is one of the things that has led to some of these long-term durable responses that we're seeing in previously intractable cancers. It's obviously something that's systemic. It treats anything in the body, so that your immune system is there protecting you all the time, so we can capitalize on that. It's very synergistic. It can be used in combination with a lot of other standard therapies or experimental therapies. It's also targeted in terms of you can really focus the immune system on a specific antigenic target within a tumor, so that also makes it a very appealing thing. And I think it's universal in that, again, it could be applicable to all types of cancer. So that's a very quick overview of the major characteristics of immunotherapy and why it's important. I think you all know that in the last, I guess, basically 7 or so years, there has been tremendous progress in the number of FDA-approved clinical cancer immunotherapies. The majority of these, as highlighted in red, are checkpoint blockades, which really take the brakes off the immune system and allow the immune system to continue to function in an immune response. But there are other types of immunotherapy besides checkpoint. We've also had 2 approvals of CAR T cells, which are genetically engineered T cells from patients that are given back a very personalized treatment. But there's also been bispecific antibodies approved, as well as oncolytic viruses. So that, this amazing progress that we're seeing, I think, probably, I guess, there's been about 16 different immunotherapies approved for about 15 different types of cancers in this last 5 to 10 year period. So it's an exciting area. And I think we finally have proof of principle. I think many clinicians that would have doubted that the immune system could be used to treat cancer have been kind of put to rest, that doubt. I think at least in the right situation, in the right cancer patient, we can see that immunotherapies can be effective. But, we still have a long way to go. This is just a very, very simplistic term saying that obviously showing survival if there's no therapy. What conventional, being radiation, chemotherapy, has done, it moves the time along in terms of giving patients a little bit more time, but many still end up dying. And I think cancer immunotherapies are exciting because they're raising that tail of the curve, and you're seeing this long-term durable responses. I mean, it's really almost miraculous in patients where some of these original checkpoints have worked, that they're now 10, 11 years out. People that were probably 6 months close to dying are now alive and well after having been treated only once, through a regimen back then. So, but the problem is, although is all the excitement there is in cancer immunotherapy, this is not a complete response for every patient or for every cancer type. So we still have a lot more research to do, and I think the world would agree that it's combinations, figuring out what combinations, how you can apply cancer immunotherapies with different types of cancer immunotherapies or with other treatments where we can continue to raise this tail of the curve and hopefully be more effective for more cancer patients, which is the ultimate goal. So this is kind of just understanding that just like focused ultrasound, which has 18 different mechanisms of working, the immune system is a very complicated system. And so it isn't just a one mechanism that takes place. This is kind of showing you kind of the immunity cycle, that there's about 6 different steps that have to happen to get an effective immune response against cancer. You need, for the first 1, 2, and 3, you really need to generate cancer-specific T cells, and this takes a number of steps. You then have to have these T cells traffic to the tumor. Then they have to infiltrate the tumor site and then deal with immunosuppressive that's naturally there as the tumor tries to protect itself from an immune response. So I've kind of put this together. It's a complicated style, but at every one of these steps, there's obviously things that can either positively or negatively affect those steps. And I've tried to highlight it, and I don't know, didn't come out, but I did try. Can you see? I can't see it in this, but maybe there are in red. I kind of put areas where perhaps focused ultrasound could be used at these various steps, perhaps in generating cancer-specific T cells. In fact, it could be in terms of, dealing with what cytokines are actually produced because of treatment with focused ultrasound, or it could also be using, as we've heard already, disruptive in terms of breaking down stroma that allows T cells to get in, or like the blood-brain barrier, so things can get there. So I think these are open questions. I think we don't know how focused ultrasound really affects the immune system in detail yet. I think we're getting there. And then, of course, how would you combine this? So I think just to put this a little bit more in perspective, this just shows one of the things the Cancer Research Institute has done over the last 5 years is really tried to track the field of immunotherapies. And this just is it's another complicated slide, but each column is a different checkpoint checkpoint blockade approved therapy. So you have pembrolizumab, nivolumab, so there's 5 of those, and then there's others that have not yet, PD-1s. These are just PD-1 checkpoint blockades. And this is showing since September of 2017 as compared to June of 2018, the number of clinical trials ongoing using one or another type of anti-PD-1 or anti-PD-L1 therapy has risen from about little over 1,500 to over 2,000. So this is a very crowded space. And I think I've heard cancer immunologists say that it's almost like throwing spaghetti at the wall right now and seeing what sticks. Take whatever you have and combine it with an anti-PD-1 or PD-L1. And I think our thinking is we need to take a step back from that. We really need to understand mechanism. So though there's an opportunity here with all these trials out there, we need to get samples and we need to really interrogate them in a very precise, with top-notch immunological assays, which change all the time. So you really need people that really know what they're doing, measuring in patients what's actually happening here. And then I think you also have to really fund the very basic research to understand the mechanisms of how this works. So I think there's no difference in that mindset for cancer immunotherapy than there is with focused ultrasound. And I think that's what needs to take place. And because of that, we've been very lucky that we've, the last, I guess it's now, 4 or 5 years, I suppose, that CRI has been partnering with the focused ultrasound to see, is there a role for focused ultrasound and immunotherapy? We started with some workshops, we've done scientific meetings, we're now co-funding some of this. And I think the important thing about the co-funding is cancer immunologists don't know about focused ultrasound. And focused ultrasound people don't know about immunology. So I think the key thing is to bring expert scientists from both sides together, and that's what we've tried to do with our co-funding, pairing people together so that we can get the most that we can out of every early clinical trial, or even any preclinical or mouse model in terms of what is happening immunologically with focused ultrasound. Because I think only with that knowledge can we really intelligently really bring this to the clinic in the most defined way that we hope to get the most out of it. So I think that's kind of where I leave things here, and I think it opens up to Kathy, who's really more of an expert in this field than me, so she can bring you on to the next point.
Great, thanks so much. So I'm going to tell you a little bit about some of the mechanisms by which we think that focused ultrasound and immunotherapy can be synergistic. So part of the idea is that we know that one of the problems is that our body is not recognizing a tumor, for example, that we'd like to educate the immune system, and we'd like to be able to teach immune cells to go after a cancer. So if we had a multi-site cancer, if we were to add focused ultrasound to that, and you've heard from Neil and from others that ultrasound has the opportunity to go in and ablate or apply heat to a tiny little region of a tumor. It also has the ability to mechanically alter effects. So if we take those sort of effects, can we accomplish something really miraculous in the sense of being able to educate these immune cells and then have these immune cells be able to travel and find other sites of disease in other regions of the body? So that's our goal. And what I'm going to tell you is that we find that there is a synergism and that we can combine these things in different ways. And I'm going to try to tell you a little bit about why we think that's the case. So one mechanism by which we think that there is a synergy is that we do see that a thermal ablation, when we've applied heat to a region and we have slowly dying tumor cells surrounding that ablated region, we see that the tumor is sloughing off some of its native proteins. Some of the proteins that are unique to that tumor are being sent out into the surrounding tissue, and they're being picked up. And they're being picked up by macrophages and dendritic cells and then displayed to help activate your immune system. So this is one of the reasons. And this is a little bit of data to just indicate that this is one of the things that happens. So we have a very specific, it's a chicken ova albumin that can be transfected into a tumor and can be displayed. So it's something very unique that we're able to look at. We can make that happen, and then we can go ahead and we can ablate particular tumor, and then we can look systemically and we can look locally and see, is that specific antigen presented by immune cells? And we find that within the tumor, the graph on the left is showing you that within the tumor itself, it is specifically presented. In the surrounding lymphatics by itself, we find that there's not a lot. So we stepped back and said, how can we make this better? And what we find as a mechanism or as one way of improving this combination is to provide a priming regimen of immunotherapy that includes checkpoint inhibitors, and in our case also includes an adjuvant to rev up the immune system ahead of ablation. And part of the reason that this is an interesting and important aspect of things is because the cell death that occurs as a result of ablation is very, very fast. And so we want to get the immune system prepared. We want to have the right population of macrophages and dendritic cells there ready to see those proteins when they're sloughed off and be able to take them and be able to respond with them well. And so what you see on the right-hand side then is a graph. We're using the notation AI for a combination of ablation and immunotherapy. And we've got lots of, in the lymph nodes, we've got lots of the specific antigen being presented on cells, on our desirable, in this case I'm showing you just a combination of CD45 positive cells. But by best case, if we combine ablation with immunotherapy, we're seeing a lot of that specific tumor antigen being presented and being available to activate the immune system as a result of that combination. So that's one of the mechanisms. And so this ova-albumin is now being detected in the lymph nodes, and this has really been enhanced by ablation, we find. But the effects are bigger than this by this combination. We find that there's in total a combination of about 10,000 genes that are whose expression is changed by the combination of immunotherapy and ablation. And that's shown in the Venn diagram on the left there. And that's in the treated tumor, about 7,000 in distant tumors. So again, the idea is that we're only applying the ultrasound to one lesion, and then we're looking at gene expression even in very distant tumors. And we see that there might be 7,000 genes that are altered in those distant tumors as a result of this local treatment. The histology that you're seeing on the right-hand side of the slide is also showing the AIC, again, as a combination of ablation and immunotherapy, and the -C means in a distant tumor. So in a distant tumor, we're also seeing a much greater therapeutic effect as a result of this combination of these ablation plus immunotherapy coming together. So from this slide, I'm really telling you that we see this anti-tumor gene expression being enhanced, and also we see this anti-tumor efficacy that we're looking for being enhanced by this combination. So then I'm going to take you through two specific aspects. So what we found is that things are very different in the local tumor environment that we treated and in the distant tumor. And so we've really gone back and tried to characterize this. If we really focus in on what are some of these 10,000 genes that are changed in the local tumor, many of them have to do with upregulated genes as a result of type 1 interferon. These are like a virus. So it's as though your body has seen a virus there locally, in part because we're sending out extracellular DNA that's being taken up by your immune cells. So your body is responding to that as though it's actually seeing a virus there locally. And we see in the treated tumor, we see a substantial upregulation of these viral-like gene expression patterns. One of the interesting things is that this has also been seen in radiation therapy. So when you're looking at how to combine some sort of focal therapy, be it radiation or be it focused ultrasound, in each case, one of the things that we see is this combination of genes that are associated with a viral response that's helping to get your immune system up and going. As a result, in the distant tumors, what we see are your cytotoxic T cells. So these are the T cells that are often thought of to actually create a lot of the anti-tumor effects. They're going in and they're able to generate even more chemicals to help kill tumors, and they're able to help to clear the site. So when we look at the genes that are changed in the distant lesions, we find that they mostly are T-cell related genes. And it's both the number of T-cells, the little red arrows are showing you that in the distant sites as a result of ablation and immunotherapy, T-cells are upregulated and they're also more activated. So some of the activation factors that we would look at, like ICOS, are activated in the distant tumors as a result of the combination of ablation and immunotherapy. And in the brown histology or immunohistochemistry on the right, you can see the little brown spots. Those are all T cells. Those are all CD8 T cells in this particular case. And they have invaded in a very high density into that region and really kind of taken over that distant lesion to help to clear it. So this is, again, another mechanism by which this combination is acting. So just in summary, what I'm communicating is the idea that we're intervening locally with this ablative therapy, and we're combining it with immunotherapy. Our adjuvant is also administered locally. We're combining that with a checkpoint inhibitor that is a systemic effect. But we find that this combination can be much more effective in upregulating the immune system and changing both your innate immune cells, macrophages and dendritic cells, but also your T cells. So thank you very much.
Good afternoon, everybody. My name is Dr. David Brennan, and I've been tasked with giving you a preview of some of the sessions that will be coming over the next few days in this meeting. I'm going to be presenting data from other investigators mostly on the intersection of cancer immune therapy and focused ultrasound. But before I do that, I want to drive home this primary concept here. And you've heard a little bit about this from some of the previous presenters today, and that's the concept of immune privilege. And what I mean by that is that many cancers have immune privilege. Specifically, pancreatic cancer, glioblastoma, and breast cancer have more immune privilege than others. In other words, they're able to run under the radar of the immune system and evade immune surveillance by various tricks. And since Halloween is just around the corner, you're going to have to indulge me a little bit on this tumor trickery. So one of the first tricks that the tumors can use is to evade immune surveillance is they can secrete type 1 interferon. They can also recruit myeloid-derived suppressor cells and T regulatory cells. And all of these things tend to tamp down the immune response, even if there is an immune response mounted from the host to the tumor. Another trick that the tumors can use is they can express programmed cell death ligand 1 or PD1, PDL1. You've heard a lot about this already, and that's depicted here in these PDL1 protein skeletons decorating the surface of a tumor pumpkin, fighting off an immune response. And in the case of a tumor in the brain, the tumor has the ability to hide behind the blood-brain barrier. So there is a lot of tumor trickery, but hopefully we have just the right treat for these tumors in focused ultrasound, because we know that focused ultrasound can break down the blood-brain barrier and cause a local tissue injury to induce an immune response, just like Dr. Ferrara had spoken about already. And then we hope to ramp up that local immune response with various immune potentiators, such as anti-Treg antibodies, as well as anti-PD1 antibodies, such as pembrolizumab. And hopefully we can use these immune potentiators to interfere with the tumor trickery to negate the tumor immune privilege, allowing the host, be it a mouse or a human, to mount an immune response to the tumor and make it go away. So let's start off by talking about pancreatic cancer. Petros Meritidis and Gail Tahar at the Institutes of Cancer Research in London and Sutton in the UK will be presenting their data looking at focused ultrasound to treat pancreatic cancer on Tuesday at 1:49 in the afternoon. Pancreatic cancer has immune privilege in one unique way. It can produce a dense stromal reaction within the host's pancreas, and that keeps the blood supply away, keeping the immune cells at arm's length. It's also able to recruit CTLA-4 positive T regulatory cells, as well as express programmed cell death ligand or PDL1. Dr. Tahar will talk about her preclinical study in mice in which she took immune-competent mice and implanted orthotopic KPC tumor cell line pancreatic tumors into the pancreas of the mice. She then allowed those engraft tumors to grow and subjected the tumors to pulsed focused ultrasound to induce cavitation, and also gave the mice an immune checkpoint inhibitor cocktail consisting of anti-CTLA-4 and anti-PD-1 antibodies. They did the focused ultrasound, gave the antibody or the cocktail before and at several times after the focused ultrasound, and what they found is that at 12 days there was a substantial reduction in growth shown in the treated animals, shown in that blue line at the bottom in the panel to your left. And survival was also increased in the animals that received focused ultrasound to their pancreatic tumors and that immune cocktail, leading her to conclude that focused ultrasound combined with immune checkpoint inhibitors controlled tumor growth and extended survival in these treated mice. Moving to glioblastoma, you've heard a lot about this already, but glioblastoma is immune privileged, mainly because it exists in the brain, behind the blood-brain barrier, which as you already heard about, has low permeability to immune cells, as well as to chemotherapy. Histotripsy, which is a form of ultrasound, a focused ultrasound energy, can produce cavitation, which disrupts the blood-brain barrier, and also can disrupt the membranes, the cell membranes of the tumor, but can do so in a way that doesn't lead to denaturization of the protein and in fact exposes tumor antigens, hopefully resulting in the host mounting an immune response to the tumor. Dr. Gerhardsen and colleagues at the University of Michigan will present their data on Tuesday at 11:17 in the morning. There's a preclinical trial involving mice. 27 mice were inoculated with glioblastoma, and approximately 2 weeks later, half of the mice were treated with histotripsy. This is a slide that shows the experiments as they progress. On the left, you can see the tumor inoculation. They then let the tumor grow for 2 weeks, and then histotripsy is applied to this tumor in the skull. They wait 1 week, imaging all along the way, and euthanize the animals and collect tumor tissue. On the left, you have the control tumor. It's a healthy-looking tumor in an untreated mouse. On the right, this is a section taken adjacent to the treated area, and as you can see, there's lots of edema. The cells are pyknotic. On the right side, these cells are not healthy. Hopefully, they are dying. They also identified a substantial reduction in myeloid-derived suppressor cells in the treated tissue, both in the brain, the spleen, and the tumor-draining lymph nodes, and a substantial increase in interferon gamma in the treated tissue, leading them to conclude that changes in the immunomodulatory environment and tumor histology may indicate increased immunogenicity and anti-tumor responses in the treated mice. Shabani and co-workers at UVA will present their data also involving treatment of glioblastoma with focused ultrasound on Tuesday as well in the morning at 11:24. They applied focused ultrasound to intracranial implanted glioblastomas in mice after the intravenous injection of microbubbles, and microbubbles are known to enhance the effect of focused ultrasound. Then collected the brain tissue 1 week later and found in the treated mice that there was a significant increase in CD4 positive T cells, or T helper cells, and CD8 positive T cells, cytotoxic T cells. The cytotoxic T cells, for those in the room who aren't aware, are really one of the effector cells of the immune system. It is these cells that if you can get them to the tumor, sensitize them to the tumor, that they will kill tumor cells. They also found in their treated animals that they were able to inhibit growth of the tumor, as depicted on the graph on the right side. So Shabani concluded that these findings demonstrate that the blood-brain barrier and blood-tumor barrier disruption with focused ultrasound and microbubbles can potentiate anti-tumor immunity against glioblastoma in mice. So this brings us to breast cancer, which is my main field of interest. We know that breast cancer can evade immune surveillance by producing type 1 interferon. It's also able to recruit myeloid-derived suppressor cells as well as T regulatory cells to tamp down that immune response. And many breast cancers can express programmed cell death ligand 1 or PDL1, again, thwarting any ongoing immune response from those CD8 positive T cells. Dylan and coworkers, including myself at the University of Virginia, will present their work on Wednesday at 11 a.m. This clinical trial is focused ultrasound ablation of breast cancers in combination with pembrolizumab. Patients in this trial have stage 4 breast cancer, and this is truly the first of its kind in human study pairing focused ultrasound ablation with a PD-1 checkpoint inhibitor to treat breast cancer. Our hypothesis is threefold. First, that focused ultrasound plus pembrolizumab will alter the T cell and myeloid cell responses at the local site of disease. Second, that the combined therapy will result in lymphocytic-mediated immune response at distant, non-ablated sites. And lastly, that focused ultrasound plus pembrolizumab will result in an increased T cell specific immune response in the blood that will be measurable, indicating a systemic response. We're using the EchoPulse ultrasound-guided focused ultrasound device that's manufactured by Theracleon. This ultrasound-guided device we are using to partially ablate the tumor. We are intentionally leaving at least half of the tumor untreated, and then the patients are divided into 2 arms. In the first arm, Arm A, patients receive pembrolizumab after focused ultrasound ablation, and in Arm B, they start their pembrolizumab prior to ablation. 22 days after ablation, the first step is to obtain 2 biopsies, one of the ablated area and the other in the peri-ablated zone. This is going to be difficult to go through without a laser, and unfortunately, I think the laser doesn't work on the TV. So for purposes of clarity, I'm going to divide this into 3 columns and 5 rows. So the 3 columns are the biopsy prior to treatment, and the second 2 columns are the biopsy in the ablated area that I showed you before on that ultrasound, and the peri-ablated area. Those biopsies are 22 days after treatment. The next 2 rows are staining for CD8 cells. CD8 positive cells, those are the CD8 positive T cells, the cytotoxic T cells, and you can see in that first row prior to treatment in this first patient on the study, there were very few CD8 positive T cells. In the middle row, in the ablated area, the tumor's dead. There really isn't much of an immune response there, but in the peri-ablated area, there's a significant increase in the number of CD8 positive staining T cells. Remember, those are the cytotoxic T cells. Those are the cells that should be able to, if you can get them to traffic to the tumor and function there, be cytotoxic to the tumor. The FOXP3 is the next row. That's a marker for T regulatory cells. If there are a lot of T regulatory cells there, it might be able to tamp down an immune response. There weren't many FOXP3 positive T cells there, or cells there prior. There aren't after treatment. And what was very interesting to me, anyway, as a non-immunologist, was that the last row there, the PDL1 row, this tumor was essentially PDL1 negative or very minimally reactive, and it was able to mount a PDL1 response to create PDL1 protein after treatment. This was either in direct response to the ablation or perhaps more likely in response to the fact that the CD8 positive T cells moved in and were trying to kill it. So clearly, at least in this first patient, the local immune response is encouraging and mirrors what Dr. Farrar is seeing in mice. So hopefully it will result in an improved clinical outcome through abscopal effects. To date, the study is currently open, accruing patients, and 5 patients have been enrolled to date. Thank you for your attention.
So thank you to those presenters, and now we have some time for discussion. So again, if there are any questions from the audience, please raise your hand. Do we have any online questions?
We have a question about the work that Dr. Brennan was just talking about with the patients in the breast cancer immunotherapy study. They're asking how many of the first 5 patients are demonstrating an immune response, and any insight as to whether the approach with the treatment with the focused ultrasound and the immunotherapy is better than either of those treatments alone, and when will the study be complete?
So before I answer that, you need to remember that this is an ongoing trial that involves a drug and a device from 2 publicly traded companies. So I can really not talk too much about an ongoing trial, but as you can see from that panel of immunohistochemistry slides, at least on that first patient, that's already been discussed, so I can talk a little bit more about it. We've certainly set up that first hypothesis seems to be working, that we're able to induce changes in the local immune response to the tumor. Remember that the drug that we're giving is specifically designed to block that PD-1 protein and allow those CD8 positive T cells to be efficacious at the tumor. So far, so good. The study is designed to go on for 3 months for each patient. We've accrued 5. We will continue to accrue these patients, and we have several others that will be treated in December. I suspect it will take another 8 months to a year to complete accrual, but remember, it only takes 3 months of follow-up. It's our hope that the combined therapy, that's certainly our hypothesis, will be more efficacious than each one individually, and that's probably as much as I can answer that question.
Thank you.
Any other questions from the?
Not at this time.
Okay. And Dr. Bullock, I'd like to bring you in. I know you're also at University of Virginia, and you're involved in this study and many others. And so wonder if you just had thoughts on, again, in this early clinical trial, what are some of the key things that we're looking, key questions we're looking to answer, and then moving forward with future clinical trials, again, some of the things you think we really need to answer before we know what we need to know about focused ultrasound.
Wow, crystal ball time. So what we are learning from this trial, this formative trial, is the extent to which this one regimen of focused ultrasound can be impactful on the immune response to a tumor that generally has not been responsive to immune therapy to this point. The critical element to go forward is that, as Jill described earlier, is bringing in the right array of techniques that allow us to examine what's going on in the tumor, because one of the things that have become really apparent in patients who are treated with immunotherapy is that in many cases a resistance pathway becomes activated, as perhaps illustrated with the increase in PD-L1 expression in these tumors. And so Newton didn't realize he was an immunologist, but because for every immunological reaction there is an alternative response by the tumor, and so we have to be able to anticipate how the tumor responds to these insults, and the clinicians will have to be very adaptive to help bring in different counter therapies to every dance that the immune response and the tumor have together. I think the basic studies that we're doing in preclinical models should provide us a lot of insights and will help give us the armamentarium that the clinicians will need to be able to bring focused ultrasound to its fullest capabilities in these sort of patient populations. I think it's going to vary tremendously from disease to disease, which is the appropriate ultrasound regimen, because the barriers, as described by Dr. Brennan and Dr. Ferrara, can be quite discrete between the different disease types, and there are different arrays of problems within each diseases. I could illustrate this with perhaps a difference between GBM and melanoma, for example, where Dr. Ferrara is showing some very potent immune responses against the melanoma models. But in GBM, we don't have an interaction between the immune system and the tumor until the blood-brain barrier is opened. And this could be an opportunity now for the immune system to say, hey, we have got a new place to party here, and we'll be able to recognize and respond to those tumors. So as we go forward, I think we need to do the sort of approaches that are currently in place, which is to dig deeper into understanding what happens to the immune system and the tumor based on the energy deposition that is engendered by ultrasound, and then what are the adaptations that occur as a response to that.
Thank you. I'd like to bring Dr. Wong into the discussion. Being a clinician who treats patients with a variety of different tumors, I want your thoughts on where you think focused ultrasound could potentially play a significant role, maybe the specific disease area, and what you think are the next steps in that area.
Yeah, like Neil said in his introduction, I think that focused ultrasound has just enormous potential in treating a variety diseases, particularly in the cancer area. My area of research really focuses on pancreatic cancer, and one of the reasons that I focused on this area is that really, up to this date, there have been essentially no highly effective therapies, and really for the first time, I think we're getting some glimmer of hope, realizing that it will be multimodality therapy, hopefully using HIFU or focused ultrasound to help potentiate or elicit an immune response, and using some of these immune modulating agents to accelerate that. There's always been kind of these case reports of abscopal effects from focused ultrasound therapy, dating back over 20 years. And we always postulated that this was an immune-mediated response, but no one really understood what any of the mechanisms were. For these meetings that have been going over the past couple of years, we're really starting to get an idea on what the actual targets are. And so I think that the next few years are going to be incredibly exciting in terms of the growth. And I for the first time, I really do have hope that we're going to make a major dent, especially in pancreatic cancer, in terms of overall survival. And I don't think any of us had this hope, prior to this specific therapy.
Great. So drawing from, I know radiation was mentioned, and there has been a lot of work in recent years combining radiation with immunotherapy. So I wonder if any of you want to speak to kind of what we found there that could perhaps be applied to focused ultrasound, and then also, stressing the unique capabilities of focused ultrasound, being able to do more than just ablation, and how that might fit in. So does anyone want to talk about that?
Well, I can talk a little bit about the mechanism, and as Neil mentioned, there are 18 different mechanisms that we can think of in terms of ultrasound, and there are some that are just truly unique to ultrasound, and it really are the mechanical effects. There are no other interventions that I know of clinically that produces some of the mechanical effects that we see from ultrasound that are purely non-invasive. So without any incision, without, that we can just go either transcutaneously or even, in my case, endoscopically. That's what's really unique about this, and it has rational effects that will lead to biologic effects that we can really tie into mechanisms that will affect the immune response. And so I think that, especially cavitation. Cavitation is, we've heard that a lot. This is bubbles interacting with tissue that can either, change membrane potential or lyse membranes, leaving proteins intact. So it's really this mechanical bioeffect that I think has the greatest potential, and that is the most unique to focused ultrasound.
In addition, I guess other unique aspects are that the cell death is so fast, and there's virtually no bystander effect. So with a well-designed therapy, we can hit some specific area and really not have much of an effect on the surrounding tissue. The fact that it is so fast that we can repeat a treatment on a schedule that the clinician feels is appropriate. And also, one other thing that I didn't mention in my talk in terms of a mechanism is that the thermal effects also in the surrounding blood vessels around an ablated site, there's a hyperemia and a leaky vasculature. And we've shown that you can get an increase in nanoparticles in drug of anywhere from 5 and 50-fold surrounding that site. So there's also the opportunity to pull in adjuvants and agonists to enhance the response further. That sort of opportunity doesn't really exist with another modality.
So maybe, talking about the combining focused ultrasound with other therapies and sticking on that, we've heard some of the things that you were talking about, Kathy, and then combining focused ultrasound with checkpoint inhibitors. Are there other potential immunotherapies where we think focused ultrasound in combination may be potential?
I'll take a stab at that. There's two that really come immediately to mind. We talk about the antibodies in terms of their ability to interfere with checkpoint molecules and thus raise the level, but antibody-drug complexes are an obvious opportunity to promote tumor damage and thus activation of the immune response. And related to that, and to this whole world of antibodies, is CAR T therapies and a lot of the transgenic T cell therapies. If we can improve access of T cells to the tumors, whether that's by degrading fibrous networks that are preventing immune cells from trafficking there, or upregulating the homing receptor ligands, the sort of traffic signals that immune cells use to guide themselves into where they're supposed to be going, then we have great opportunities to increase the efficacy of, say, CAR T therapies, which to this point have not been great in solid tumors. So there's a whole world to be explored about how focused ultrasound energy can change the nature of these traffic signals, the GPS signal for the tumor for the T cells, so they could get in there. I mean, I don't know of any studies that have yet to even tackle that yet. So I think it's a very open horizon there. Very exciting.
Okay. Anyone else have anything to add with that one?
I just want to add the fact that by harnessing the immune system, we no longer are compelled to treat the entire tumor with HIFU or with focused ultrasound. And that has been one of kind of the barriers for focused ultrasound, is that we've had trouble monitoring therapy and trying to ablate the entire tumor, especially from a non-invasive standpoint. So, you know, the method that Dr. Brennan has taken to just partially treat a tumor in hopes of eliciting a systemic response, it just really opens the door to focused ultrasound therapy, because we're no longer compelled to treating the entire tumor. We only need to treat a part of the tumor to elicit a systemic response, and that, you know, that basically solves our problem with monitoring. And now we can go to low-cost methods of delivering therapy, and so this has a potential to be truly cost-effective therapy and not increase the costs of healthcare.
So yeah, I would agree with that change in the paradigm. You know, the ablation studies to date, at least in breast cancer, have gauged success by if you had 100% ablation of the tumor, which to me makes no sense. Even when our current best technique today, lumpectomy, we have positive margins really in the best of hands 20% of the time, but up to 40% of the time we leave residual disease that we can actually measure behind and have to go back and do a second operation to get it. So the key thing in my mind will be leveraging the immune response, being able to rely on other treatments, but also we're going to need to develop better imaging that can detect viable disease post-treatment. Because with focused ultrasound ablation, there is no reason that we can't retreat if we detect residual disease 3 weeks later. So I think that these will be the changes in paradigms moving forward that will make focused ultrasound ablation what I believe will become a superior ablative technology as compared to cryo or laser or RFA.
Yeah, I would just add that because of it seems to be, for me, not a focused ultrasound expert, but that the side effects are minimal with this, being it's non-invasive, but it also doesn't cause much adverse events. And the fact, that's always the question when you're doing combinatorial treatments, which is what I think the future holds. So certainly the future of immunotherapy is going to be personalized immunotherapy, and it's not one size fits all. It's going to be deciding what that patient needs to develop an effective immune response. And I think the lack of adverse events that focused ultrasound makes it a very, welcome, treatment option to do combinations with, because once you start adding more and more drugs up, your adverse events go up. But if this starts at 0, it's a really nice place to be.
Great. To tie us back to the previous session and talking about GBM, and Dr. Price had mentioned, thank you, the work of the foundation in trying to help get to some of these answers of the underlying mechanisms of focused ultrasound and how we might be able to elicit different responses if we're using different kind of modes of action of focused ultrasound. So I know that, Tim, you're involved in this consortium that we put together of many different sites where you're all using the same tumor model of glioma, and you're each looking at different focused ultrasound mechanisms of action. And we're just getting some results from there, but I wonder if, just based on what you know about what's happened in that study, some thoughts that it's brought to you.
Yeah, so it's early stages yet, but the intent behind the study was to have some bookends on the way that ultrasound is delivered and assessed in a very similar model, so that we can begin to get some well-controlled studies that would allow us to understand how these different regimens are ultimately impacting on the tumor, but particularly what are the correlating immune responses that are occurring in this space, so that ultimately we can improve upon those therapeutic choices by coming in with combinatorial therapies. And I think one of the things that is striking to me is, and as I say, the data is very fresh, is how there can be different degrees of immune responses just from these different regimens. So we've heard that ultrasound can be given in sort of in broad brushstrokes, either by heat or by mechanical damage, whether it's thermal ablation or histotripsy or microbubbles. And some of the early data that's coming out of there suggesting that these different regimens can have different impacts on different cell types with immune response. Some regimens seem to be promoting the elaboration of a cytokine that we know is really important for controlling tumors, interferon gamma. We know that it seems to be that some of the regimens are removing immunosuppressive cells out of the tumor, which suggests that they would again help support the type of immune response that would control the tumor's growth. But as some of the other approaches seem to be increasing some of these resistant cells, which speaks to the Newtonian side of the immunology, that if you're doing some damage, the body is actually designed to come in and get rid of that damage and reduce inflammation. And so these coordinated studies, when we're using the very similar analytical techniques, allows us to have a high degree of confidence that we can determine some of the effects of some of these energy regimens and how the immune system is going to respond to it. And this is exactly where we need to start to have a very coordinated, precise approach to say what works, what doesn't work, and where can we improve upon. And I think we have to take very similar approaches in patients going forward.
Does anyone else have anything to add? I don't know, Kathy, from your work, if you have anything to add just about the, the different modes of action of focused ultrasound and kind of next steps we need to take there.
So I agree that it's it definitely is interesting to look at the different mechanisms. So far from our own work and doing direct comparisons, we found, at least in combination with agonists, that we get a stronger CD8 T cell response in distant sites from a thermal effect. But I think there's still lots of opportunities to look at different combinations. One other thing I was just going to mention quickly is that to the comment about combinations as well, is that it's much easier to combine focused ultrasound with chemotherapy as well in many cases. So one of the most challenging aspects of the combination therapies is how to stage and how to order and how to bring things together. And so we have, because we don't damage systemic circulating T cells or circulating immune cells, there's this great opportunity to really bring things together in different ways than you could with radiation, for example. So.
And I'd just like to, again, mention that, we have established a really, I think, wonderful partnership with the Cancer Research Institute. And, moving forward, we see that partnership in partnership with some other organizations as a vehicle by which we can answer a lot of these questions that we still need to answer about focused ultrasound to hopefully drive more clinical trials of combination approaches. And so as I was asking, just like asking the previous panel about, obstacles that you see or big areas that you see that still need to be addressed in this field, that perhaps the foundation in collaboration with CRI could help kind of break down some of those barriers. I'd just like to open up to the panel your thoughts on where we should be going next.
I'd love to start with that. I just want to express my appreciation to the foundation, because really without the foundation we wouldn't be here talking about this. There are so many different players. For example, in pancreatic cancer, we're talking about oncologists, we're talking about the focused ultrasound technology community, surgeons, radiation oncologists, now the immunologists. One thing that the Focused Ultrasound Foundation is going to be doing in February is we're in the process of organizing a workshop on treatment of pancreatic cancer using all these various modalities. And this just wouldn't happen without the support of the foundation. And we're going to get everybody who's involved. It'll be a large international meeting, and hopefully from there, we'll have more ideas on how to move forward, develop closer collaborations, and certainly the foundation helps in huge ways in getting funding, increasing awareness of what we do, and helping us move this field forward. So I definitely want to express my appreciation to the foundation for everything that you've done for this field.
Yeah, myself as well. And I guess I see one great opportunity because we have such compelling data at this point. I think the opportunities for some of these first-in-human studies of combinations are, that we see from some of our colleagues at UVA and opportunities that are presenting themselves at this point. I think that you're in a unique position to help us move the field into, move many of the combinations and unique assays into human studies. So I think that that's a great opportunity, and you guys play a major role in making that happen. Yes?
I think I might want to add here that the place where the Foundation and the Cancer Research Institute has placed itself in supporting these early stage studies is incredibly important for what I consider the barrier of inertia and the NIH, that we're blessed that we have great funding from the National Institutes of Health, but it's a very conservative organization, and so having the opportunity to develop the sort of early stage studies as proof of principle and proof of efficacy allows us to be in a much stronger position to convince our colleagues who wouldn't be able to recognize a megapascal from a megapeta to align these technologies and to educate them so that they know what's coming down the pipe is it's a profound opportunity. But staying focused, and that's the hard thing in a brave new world like this, where there is so many potential opportunities and modalities in place. Staying focused on the prize, which is determining this relationship between sonic energy and the immune response, is going to be a big challenge.
All right. Well, if that's it, and no more questions, then I will close this out. Thank you all for joining us today.